Yes. Yes, so guys, today we will be doing an extension of autosomal linkage, which is crossing over. But before I move on to the topic of crossing over, let's just recap what we did last time, because there's a long gap as we did not have a class on Friday. So as far as autosomal linkage is concerned, we had actually, let me go back and show you all the crosses. So these were the crosses that we did, right? And when we were doing autosomal linkage, we said that we are also concerned with two genes on uh, having two pairs of contrasting characteristics, but the difference is that here the genes are on the same chromosome, they are linked. And since they were linked, so we put them into brackets. And now if I just sum up, what we did last time was that if it is autosomal linkage, okay, if these genes are linked, of course, they will never be written like this as they used to be in a typical dihybrid, right? So on the right hand side, it's showing you the results of a typical dihybrid in case we make a cross between two homozygous organisms. And on the left hand side, you see the same cross between two pairs of contrasting characteristics. But here, instead of writing capital E, capital E, capital A, we have written capital E, capital A, and capital E, capital A, meaning that on one chromosome like this, if I draw a chromosome, and if I say that this is a pair of homologous chromosome, instead of having these alleles like this, right, these alleles are found like this. So of course, when these will separate during independent assortment, capital E and capital A will never separate because they are carried on the same chromosome. So in that case, we did last time that if we have a cross between two homozygous organisms which show autosomal linkage, of course in F1, since capital A and capital A are dominant characteristics, they will always appear as a phenotype in the F1 generation. But if we take an F1 offspring, and if we do a test cross between an F1 offspring, which is heterozygous, and with a homozygous recessive, so instead of getting a typical dihybrid ratio of 1 is to 1 is to 1 is to 1, we get a ratio of 1 is to 1 only, which you can see over here, that we get one of this organism, which is heterozygous, and we get another organism, which is homozygous recessive, which is actually a ratio of 1 is to 1. So having said that, we had made some conclusions, okay? And these were the conclusions, which were that since this is not following the typical dihybrid ratio, so we don't get the typical ratio of 1 is to 1 is to 1 is to 1, and we get a ratio of 1 is to 1 only. So this means that instead of the dihybrid ratio, we got a monohybrid ratio, right? And the reason why a dihybrid example is behaving like a monohybrid cross is that linkage has occurred. And the alleles that went into the cross together, they remained together, which is why we did not get the typical ratios. But you have to remember that this will only be possible in case if there is total linkage, right? Which means that you have a pair of chromosomes, okay? And if let's say total linkage takes place, meaning, let's say if I have it like this, and on the other chromosome, it's small a, and small a. Now, in this case, of course, if these homologous chromosomes, they will separate during meiosis 1 and meiosis 2, the gamete that I am forming in this scenario will always be this, and in this scenario will always be this, which means that I will not be able to separate these alleles even when meiosis 2 is complete. But this will only be possible if total linkage is occurring. But total linkage is very rare. Why? Because crossing over takes place during prophase 1. And you all know that crossing over always takes place between, not between the sister chromatids. Like for example, I would say that these and these are sister chromatids and these are sister chromatids. Crossing over will take place between non-sister chromatids like this. Since crossing over takes place most of the time, so we say that total linkage is rare. That's why it's written over here. I'm going to underline this. Total linkage is rare. So then what happens? As total linkage is rare and as crossing over is a common phenomenon, so it is crossing over that will lead to the breakage of the linkage groups. Okay, they will be broken apart. It will lead to reshuffling of the alleles, hence resulting in formation of new recombinants. Since we are doing the example of Drosophila, so I can tell you this, that in male Drosophila, crossing over does not occur. So since in male Drosophila, the crossing over does not occur, you might see the total linkage has happened. Whereas in all the other cases, whereas in female Drosophila and the other organisms, the crossing over is a very common phenomenon. So hence, the exact 1:1 ratio that we got over here, this is the 1:1 ratio. This 1:1 ratio that we got over here is not such a common occurrence. We might get some different ratios. So let's see what is the effect of crossing over on autosomal linkage. I'll go to this slide to show you the effect. Yes, the same example of the Drosophila fly and the same characteristics. Now you have to look at this cross very carefully. On the right hand side over here, you see a male ebony body Aristopedia antony Drosophila. And on the left hand side, you see a female striped body normal antony. Right? Acha. Now what happens over here is, and this is the heterozygous, okay? This is the heterozygous female and this is the homozygous male. Acha. We know from our prior knowledge that in the male flies, crossing over never happens. Okay, so if crossing over is not happening, it means that total linkage will happen and the gametes that will result will always be of one variety, right? There will be no crossing over or no exchange of alleles over here. Acha. In female fly, which is striped body normal antony, as you can see over here, capital E, capital A, small a, this shows when I put them into brackets, this shows that these alleles are linked, okay? These genes are linked. And over here, the crossing over is occurring. Now there is no crossing over here. And hence, when we have gametes, they will all be written like this. On this side, the crossing over is taking place. And you can see two scenarios, okay? This is the scenario. Let me write it like this. This is the scenario 1, and this is scenario 2. Now, scenario 1 is the one in which no crossing over is taking place. No crossing over is taking place. And over here, crossing over is taking place. Okay, so you can see that when no crossing over was taking place, okay, there was no connection between the two non-sister chromatids, no breakage of linkage groups. The linkage groups are intact. You can see over here, they're not breaking. And so when meiosis 1 and meiosis 2 happens to occur in this scenario, you will either have gametes which are capital E, capital A, or you will have gametes which are small a, which is very, very obvious in case of a heterozygous organism. Look very carefully at this diagram and you will see that crossing over has taken place. Okay, now let me zoom into this to show you what's happening actually. capital E, capital A, it was like this. See, capital E, capital A, capital E, capital A. Now there is crossing over, means that this allele will break apart from this allele. Okay, this linkage group is broken. The linkage group of capital E and capital A is broken. And this linkage group is now forming another linkage group. If I follow the crossing over of this allele, capital E, it is crossing over with this allele small a. So a new linkage group is made because of reshuffling of alleles, and a new recombination has been made. And what is that recombination? If we closely follow this, one recombination is capital E and small a, जो कि हमें यहाँ नजर आ रहा है. So now at the end of meiosis 2, the chromatid that I have will have a new recombination of alleles, which will be, as you can see over here, capital E and capital A. एक तो ये हो गया हमारे पास. Let's follow the other allele. This allele, small e, was making a linkage group with small a. So now as we have broken this linkage group, it will go, small e will go and form the linkage group with capital A. So the other linkage group will be small e, capital A. So this means that in the female fly, this was the female fly, right? Which was heterozygous. Because of crossing over, we have now how many kinds of alleles? Varieties. I'm saying kinds. We have 4 kinds. First kind, capital E, capital A. Second, small e, small a. Third, capital E, small a. Fourth, small e, capital A. I hope that is understandable to all. So this means that if I go back to the previous slide, I had told you that whenever crossing over takes place, we have 3 outcomes of that. Number 1, linkage groups are broken. Number 2, reshuffling of alleles is taking place. Number 3, new recombinants are formed. So as you saw, when the crossing over was taking place during non-sister chromatids, the linkage groups were broken, as we can see over here. Reshuffling of alleles is taking place and new recombinants have formed. अब ये जो recombinants आपको नजर आ रहे हैं, they were not there in the original parent. मतलब इस parent से we could either make this kind of a gamete or this kind of a gamete. अब हमारे पास बिल्कुल different तरह के एक और 2 gametes आ गए हैं. So this means that is why we say crossing over leads to genetic variation. If let's assume कि female में भी crossing over नहीं हो रही थी, तो the only variety of gametes that would have resulted would be either this or this. Now, because of crossing over, we have got genetic variation because we have now 4 different varieties of gametes. Whereas, since there was no crossing over in the male Drosophila, so the linkage groups were not broken and we only had one typical variety of gamete. अब यहाँ पे one typical variety क्यों है? Because the organism was homozygous recessive. ठीक है? अच्छा, so now if we make a cross, ठीक है, let's see के क्या cross बन सकता है. We are now, what are we doing? We want to make a cross between a heterozygous fly and a homozygous recessive fly, ठीक है, in which crossing over has taken place. Before that, we were, इससे पहले जो हमने cross किया था, वो क्या किया था कि we were crossing a female fly heterozygous with a male homozygous recessive fly in which no crossing over was taking place. So we got a ratio. पीछे हमें क्या मिला था? Ratio मिला था 1:1, right? But now if crossing over has taken place, we will get a different ratio because ये जो ये parent था हमारे पास, the heterozygous one, the female one in which crossing over had taken place, we had 4 varieties of gametes. And to this side, we only have one variety of gamete because there was no crossing over. So if we make a cross, if we cross this and cross this and this and this, you can see that we have 4 varieties of offsprings. Okay? Now, if you very closely look at the genotype of these 4 varieties of offsprings, you can see this one, let me just shade it with different colors. Let me shade it with pink. Now this pink one at the top and pink one below, I have used the same color for both. They resemble the original parent. Okay? This one resembles the female parent. Okay? The female heterozygous parent. And this one resembles the male parent. So when we get these offsprings, we have 4 different varieties. 2 are the parental varieties, इसको हम कहेंगे parental varieties. 2 are parental and 2 are recombinants. Okay, these ones are the ones in green are the recombinants. बिल्कुल new varieties, right? Sorry, I should have, I put the wrong color. This is the parental one. Let me just rub it. These are the parental ones, okay? And these ones are the recombinants. And you can see that the genotype of the recombinant is very different. Over here, you have capital E, small e, capital small e, small a. And here we have small e, capital A, and small e, small a. So these are 2 completely different varieties, okay? Which is stripe body and aristopedi antennae, this one. And the second one is a bony body and normal antennae. So ये बिल्कुल different variety hai. Now, in such case of a cross, whenever we have a cross like this, in which there is autosomal linkage also and crossing over has also take place, we will always get new recombinations, new recombinant offsprings basically, in which you have to remember one thing, okay, the parental phenotypes will always be in large numbers. ठीक है, यानी कि ये जो प्रोड्यूस हुए हैं ना ऑर्गेनिजम्स, ये वाले, दिस वन इज अ पेरेंटल फिनोटाइप एंड दिस इज अ पेरेंटल फिनोटाइप, दे विल ऑलवेज बी इन लार्ज नंबर्स, which means कि इनकी frequency ज़्यादा होगी, and the recombinant phenotypes will always be in small numbers. The reason why the parental phenotypes will be in large numbers is that you have to remember that crossing over is taking place only at one little site. ठीक है, एक जगह पे crossing over हो रही है. It's not occurring at multiple sites. यहाँ पे भी, यहाँ पे भी. Okay, so this means that if crossing over is only occurring at one little single site, the number of the recombinant phenotypes that you get will be smaller as compared to the parental phenotypes. और अगर क्रॉसिंग ओवर और भी बहुत सारे साइट्स पे हो रही हो, देयर आर मल्टीपल chiasma formation, the number of the recombinant phenotypes will increase. So this means that whenever we make a cross, हम जब भी क्रॉस करेंगे, in case of an autosomal linkage, and in case if the crossing over has also occurred, in the end, what do? We count the number of the parental and the recombinant phenotypes. अगर हमारे पास parental phenotypes बहुत large numbers में आते हैं, so this means that, and we get the recombinant phenotypes in small numbers, so this means that the crossing over has occurred, but only one chiasma maybe formation has occurred. But if the number of the recombinant phenotypes is more, then this means that there are more than one chiasma formation, और बहुत सारी जगहों पे जो है वो crossing over हुई है, right?